Hi, this is Mrs. Saul, and today we will be decoding DNA sequences to reveal a word that corresponds to an item in the classroom. It is your job to decode these sequences and snap evidence of the item using the Goose Chase app. A single DNA molecule may contain hundreds to thousands of genes. However, only the genes that are being expressed are copied into RNA at any given time. Transcription is the process of copying a base sequence from DNA to RNA. This process begins when RNA polymerase, the enzyme also used to pull apart the DNA strands, helps assemble the complementary RNA molecule using DNA as a template. The RNA is edited to only include the needed information and leaves the nucleus. During translation, the RNA is decoded into a protein. This occurs at the ribosome. Bases are read in groups of 3 called codons, and the appropriate amino acid is brought to the ribosome by the tRNA molecule. The amino acid chain continues to grow until the stop codon is read and the protein is complete. The mRNA and protein are released from the ribosome, and the mRNA strand can be translated again as cells need this protein.
Now I want to show you how to access and use the Goose Chase app for our activity on protein synthesis. First, search for the Goose Chase app in the Play Store or App Store on your device. Once you see the icon for 2 webbed feet, click Install. Once installed, you are almost ready for the activity. When prompted to do so, you will open the app in class and click Play as Guest. I will provide the game code for your class and you will enter it on this screen. An example is shown for you. Each class will have its unique code. Once you have entered that, click Submit. Now you can select team name and join the game. Create a team name that is school appropriate. I am going to choose RNA. The access code is going to be if you have a partner. Then click Create Team and Join Game. As you play, you can keep track of your team's remaining tasks and those that have been completed by toggling at the top. Use the bar graph icon to compare your score to the other teams. When you are ready to submit a response, click on the corresponding prompt, and then you are going to click on Snap Evidence. This app will require you to allow the camera usage. Once you are ready to snap your image, all you have to do is point at the image and click. You can add a comment if you like, and if you are happy with your answer, then you can hit Submit Evidence. Once you have completed all of the ribosomes in this task, you're done. Good luck, have fun, and get snapping.
Let's review the process of protein synthesis with an example. Here we have a DNA molecule with the following nucleotide sequence. The complementary mRNA strand is going to be created in the nucleus during transcription. Recall that A complements T, U complements A, C complements G, and G complements C. When transcription is complete, the strand leaves the nucleus. Now it is time for the code to be translated into a specific amino acid sequence. mRNA is read 3 bases at a time, and using the genetic code, the correct amino acid is added. This process continues until a stop codon is read and the protein is complete. Remember, amino acid order dictates what protein is made by the cell, and that code is embedded in DNA. Protein synthesis is a process that occurs in 2 stages. First, in transcription, genetic instructions are transferred from DNA to RNA. Second, during translation, the genetic code in RNA is read and used to make a protein. These 2 processes are the central dogma of molecular biology, DNA to RNA to protein. You have just modeled how DNA contains the code for the creation of specific products needed by all living things.